Hello, everyone. Our apologies for a little bit of a delay and technical difficulty getting going today. Today we have Molly Quade. She's a Washington State University graduate student studying metarhizium fungi in the Hopkins lab. However, tonight we're going to talk about honeybee instrumental insemination, which will kick us off with a little bit about some honeybee genetics, how it works, and how WSU is using instrumental insemination to bring new genetics into the US and give us some really new bees to work with. So with that, I'm going to disappear and we're going to kick it off to Molly to take it away with us.
Thanks so much, Courtney. And thanks everyone for coming out here tonight to watch this over the interweb. So like Courtney said, I usually work with fungi, developing them as biological controls, specifically for varroa mites. And if you're interested to hear more about that, I will be talking about that, presenting about that at this same event in October. So tune in for Ask a Washington State Beekeeper in October to hear more about my research with that. But tonight I will be talking about instrumental insemination and honeybee breeding and how it plays a role in commercial apiculture. Let me go ahead and start my timer so I'm talking too long. So this is a topic that I have ended up being very passionate about because I've been very lucky and had the amazing opportunity of working with Susan Kobe, who is the world expert in honeybee queen instrumental insemination. And she is now retired from working with the university, but she was a member of WSU's lab for quite a while, and she was sort of the main force behind our breeding program. And since she's retired, she's still teaching classes privately, and I've been lucky enough to be able to assist her with these classes. So I have some pictures here from some of these classes, and yeah, it's a very niche skill that plays a big role, and I'm gonna explain why. But first, in order to do that, I kind of need to give a little bit of background. So we're all familiar with the honeybee we keep here in the US, which is Apis mellifera, the Western honeybee. But we know that there's around 10 to 12 species of honeybee, and these are all in the genus Apis. And the four main most prolific species, there's Apis mellifera, of course, and then the other ones are Apis florea, the dwarf honeybee, dorsata, the giant honeybee, and then serrana, the eastern honeybee. And Apis mellifera and Apis serrana are known as cavity nesters. This is, of course, why Apis mellifera makes such a good pollinator for commercial pollination, where we're needing to move pollinators around because they like to nest in a cavity, so we can simply move their cavity around. But Apis dorsata and Apis florea, on the other hand, are open nesters. So these images here that you'll see showing this open comb just out in the open, not in a cavity, that would be an example of how they would nest naturally. And this pink area on the map here represents where Apis mellifera is native and or endemic to. So quite a wide area. And then this other area in the eastern part of the supercontinent here is where Apis dorsata, Apis florea, and Apis serrana are from. So Apis mellifera already kind of has covered a lot different ground, a lot different climates than the other three most prolific species of honeybee. And with that, they've acclimated to a wide variety of these different climates. On the lower left here, this is a picture of my boss, Steve Shepard, in Egypt with an Egyptian beekeeper. And you'll see how they have these tubes made out of mud that they're using as hives for their bees. And the ones on the top and bottom are empty to allow some airflow and some insulation from the hot sun. So this would be an example of Apis mellifera being kept in an area that's very hot, very dry. And then on the upper right here, this is actually a picture of one of our bee yards here in Pullman, where we keep some of our open mating yards. And You can see it gets really cold here, as many of you probably know and have experienced in other parts of Washington. So just keeping that in mind, the Apis mellifera is adapted to so many different climates and regions. And there are 28 recognized subspecies of Apis mellifera so far that span this area. So yeah, like all the way down in South Africa, we have, and even Madagascar, there are subspecies that are endemic to those regions. And of course, all of these are endemic to the old world. Honeybees, we don't have any native honeybees to the Americas. And when we're talking about these subspecies of Apis mellifera, if they form a Latin trinomial name, so we would refer to them as, for example, like Apis mellifera caucasica for the Caucasian honeybee, etc. But these are all subspecies of Apis mellifera, which we keep in the US. And we know that honeybees were brought over here pretty early when European settlers were coming over. And we know that they were established in Virginia by 1622. And this is quite a huge feat to have brought a colony of bees across the ocean and for them to have survived, and for that queen to have survived. Because if you think about it, they were on the ocean for weeks and weeks and weeks. And once over there, we know it was Apis mellifera, so subspecies mellifera, that was there established in Virginia. And you can imagine, because it's so difficult and risky to be bringing colonies across the ocean, once Apis mellifera was established there, that was kind of the main population for a while. And it wasn't until the 1800s when the invention of steamships came about, where other subspecies began being brought over as well. Something kind of interesting is I learned recently that apparently honeybees were nicknamed the white man's fly when they were first brought over, because they would be a signal that settlers were moving into that area, for the indigenous people to recognize. So, but that would have been Apis mellifera. And then in the 1800s, with steamships, we brought across Apis mellifera ligustica and then Apis mellifera carnica, so the Carnican honeybee. And this was all well and good. People were buying bees, and there were people keeping bees during that time in the 1800s. But right around the turn of the century, there started being this new disease people were observing, and what they were observing was honeybees were going out to forage, and then they were either seen crawling on the ground, or just seen crawling outside of their hives. So this disease was nicknamed crawling disease because they were crawling. And it was first observed on the Isle of Wight off, I believe, the southern coast of England in 1904. And it wasn't until 1919 that this was discovered to be caused by the tracheal mite. So these were mites that infest the honeybee's trachea. And of course, insects, they have trachea that are not just transporting air to their lungs like how we would, but they don't have lungs, it's transporting air throughout their whole body. So having mites in the trachea is going to prevent bees from being able to fly, because it's going to prevent that movement. So in an effort to prevent the tracheal mite from spreading, many countries began putting laws on the importation of live honeybees, and the US did this as well. So since 1922, it has been federally illegal to import live honeybees into the US. And of course, by now we know that tracheal mites are already in the US, and they're kind of a non-issue. Our bees have built up a hardiness to them, from what I understand. We never really hear about people treating for tracheal mites. However, it's still a law that makes sense to have, because there's always going to be other pests, parasites, diseases, viruses coming across if we are going to import live honeybees. So for example, right now, everyone's worried about tropicolaps. So still makes sense to keep this law in effect. And this picture here is Steve Shepard's great-grandfather, who would have been alive during this time. And Steve didn't really know him, but he said that him looking at all of his great-grandfather's equipment and everything was what originally got him started into beekeeping. So This guy sort of inadvertently is responsible for the bee program today. But yeah, so with knowing that we can't import live honeybees into the US, there people were already able to rear queens and like breed different diversity from what was still in the US. But of course, this only would have been with the grafting methods, also known as the Doolittle method. But I think probably most beekeepers are familiar with grafting, where we're simply scooping an egg from a colony that of a queen that we like, not an egg, sorry, a very young larva, transferring it to a grafting cup, and then allowing a queenless colony to build that into a new queen. However, I did misspeak earlier. It's not breeding. This is simply known as queen rearing, because in this situation, when you're grafting, you're not having any control over the paternal lineage. So you can graft and graft and graft, but you're only really able to select traits that you want from that maternal lineage of that queen that you like, because you don't know what that which drones that queen mated with, and therefore you don't know what genetics are going to be in those eggs. So grafting, prevalent, but it's a method of queen rearing. So knowing that that was occurring, but we're not able to bring any live bees in, kind of thinking about it in this large scale frame, you can imagine that no matter how many bees we have, we're always going to have this very, very limited amount of genetic diversity, because we started with this, however many amounts of colonies were brought over originally in the 16 and 1800s. So this is an example of like sort of genetic bottlenecking that we would see in nature, and in situations like this with human interaction of introducing a species. So no matter how much we select, we're still always going to be limited with whatever diversity we had initially. So how can we combat this? There's got to be a way to bring in new genetics, right? And queens, of course, are very lucrative, right? When beekeepers need to ensure that a certain fitness of their colonies, they're going to be caring about the fitness of their queen. So even as early as 1922, there are ads for queens. And other than just bringing in new genetics and wanting genetic fitness through diversity, how can we potentially bring in more genetics to maybe have different subspecies of honeybees that could be acclimated to different climates and different situations, or maybe be more hardy to certain stressors? So there's many reasons we would want to bring in new genetics to the United States. And luckily, through this method of instrumental insemination, we are able to do that, because in order to instrumentally inseminate, you have to manually collect honeybee semen. So by collecting semen in other regions or other countries, we can import that semen, do the insemination here in the US, and then have those genetics present here in live stock. And of course, this is then allowing for control over paternal lineage. So this would be considered true bee breeding. And here's a little bit more of what instrumental insemination looks like. So we have semen that's been collected into a capillary tube, and that's using a Harbo syringe, which is it has a very fine micrometer. And the semen has been collected into this tube using capillary action from saline that's loaded into that micrometer. And the queen is anesthetized using CO2, and the semen is then ejected back into her. And this might sound, I mean, it's pretty wild, but even just summarizing it as that is simplifying it a lot, because really there's a lot that goes into this. So for example, just collecting the semen, you have to go out and first collect a whole bunch of drones and then bank them, transport them. And drones, they're very fragile. They're very short-lived. They will die very quickly. So your banking has to be done in a very particular way to ensure that they will survive transport. And then, of course, the collection of semen is also probably the most time-consuming part of the whole entire process. And then on the flip side, there's just dealing with the queens. So in order to have a queen available to inseminate, you need to have virgin queens available. So that involves plenty of grafting ahead of time and rearing of virgin queens, and then banking them, and then the insemination, and then, of course, the aftercare. So here's what the manual collection of drones would look like. This is usually one of the main tasks that I'm doing when I go help Sue with her workshops is I go up and collect the drones for the students to then practice the semen collection. But in this video, you can see I'm basically just squatting in front of a colony and manually picking drones off of the entrance of the hive as they're returning from their mating flights. And of course, these drones have not mated yet, or else they would not be returning. They would have died in the process of that. But usually this can easily be done by going out from 3 to 5 p.m. That's the time when they're usually returning. And of course, that can be dependent on the weather as well. And we're using a queen excluder material over the entrance so that workers are able to go in and out of the entrance just fine for foraging, but the drones will build up on the outside. And once you have them, you would bank them, bring them to your workstation here in a very, hopefully mostly sterile workspace. And then you begin averting the drone. So here's sort of what that looks like. This is a video on YouTube of Sue averting some drones. And yeah, it's kind of a tricky motion to get the hang of, but basically involves crushing the thorax, pulling back the abdomen a bit, everting the endophallus, which is just a partial eversion, and then continually squeezing until the semen is averted all the rest of the way. And most drones, or many drones, will not yield because many drones are going to be immature, or they'll defecate on your finger and then it's no longer sterile and you got to throw that one out. So really only probably 25 to 50% of the drones that you manually collect end up actually getting their semen used up into the straw. And of course, you can kind of make this easier on yourself when you're collecting drones by feeling ahead of time, does this drone seem mature? The rule of thumb is buzzy, not fuzzy. If a drone is all fuzzy and doesn't feel buzzy at all, he's probably a young one. But if he's vibrating and has very hard tergites and is not fuzzy at all, then he'll be more likely to have a good selection of semen to collect. And here's kind of a little bit of what this instrument looks like that we're using. This is the instrument that's used for both the semen collection and the insemination. So when you're collecting the semen, you're holding the drone up to this instrument, up to the straw here with a tip, and dialing back that micrometer to use the capillary action and pull it in. And then later you're using this same setup for the insemination with these sting hooks and queen holding apparatus down here, which I'll show next. So yeah, the semen collection, we talk about it as instrumental insemination, as II, but really the semen collection is really the important part and the most time consuming, most tedious part. And one thing I forgot to mention is that everything that goes in that straw must be, for the most part, sterile, or your inseminations will not work out. Like the queen will get infected. So everything that goes in there has to be done very sterilely. So if that semen touches your finger or anything, you got to discard it and start again. And here's what that looks like, the insemination. Let's see, is it playing? Yes. So the queen is anesthetized using CO2, and she's placed in this little queen holding apparatus. And then we use this straw to go in there. So she is asleep when this happens. And then these two different hooks on either side. One is the ventral hook that sort of just hooks her and holds her open. And then we have the sting hook that actually is a little clasper that clasps the sting and pulls it the opposite way. So she's got the queen putting her in there, and then the CO2 is fed through this instrument. We have like a tube running into that holder. And you can see the queen's like breathing really heavily right now because she's still conscious and is like, what the heck's going on? But usually we see that slow down in just a little while. And then, yeah, so the ventral hook is used to just hook and pull. And then we have the sting hook that's clasping and opening her up the rest of the way. And here's what that actual footage looks like. Kind of eerie when you're not used to seeing it, but yeah. And there is kind of a bit of a method to it. You kind of got to do a zigzagging motion when you're moving the dials that move that semen straw in, because she has something called the valve fold that sort of like interrupts that area of entry. So you kind of got to go do like a zigzag motion. Kind of one of those things you kind of just have to do it to in order to like understand it. But anyway, so through this process, WSU has been able to go to other countries and collect semen from whatever subspecies they want and bring it into the US. So over the years, there's been many different trips made. And WSU is most known for importing the Caucasian subspecies of honeybee and also a couple of others. But the Caucasian one from the Caucasus Mountains is really good for areas like, for example, here in Pullman, like in that picture I showed earlier with the really hot, deep snowfall, because since they're from the Caucasus Mountains, they're very cold hardy bees. So that's something good for these colder climates. But I just love seeing these pictures of my bosses going to these different areas. So Brandon, my current advisor, he is on the right here. But this, I think, is back when he was a PhD student. And he's collecting semen from drones in, like, I believe, like a chicken hut or somewhere. Yeah. But kind of the reason this plays a key role in apiculture overall as a commercial level is because all commercial beekeepers pretty much are going to be getting their queens from commercial queen producers. And commercial queen producers are located, for the most part, in two different areas, either the South or the West in California. And with that in mind, only 473 queen mothers were used to, like, their genetics were used to replace a third of all the managed colonies in the US in at least 2004, 2005. But that's pretty standard. And so if you think about it, like having less than 500 queen mothers supply the genetics for that many bees, that's like not a very wide pool of diversity. So what happens is WSU imports this semen, and once in the US, will perform insemination. And once the queen is inseminated with those genetics, she will lay eggs, and those eggs will produce daughters with 50% of the desired genetics that she was inseminated with. Only 50%. So then what needs to be done is repeat the process, inseminate again and again and again, back and forth, until we have a daughter queen with 97% of the desired genetics we want. So after that happens, we will sell breeder queens to these commercial queen producers. And, it's not like somebody would buy an instrumentally inseminated queen and use that in like a commercial honey production. Not really, no. So the point of having the inseminated queen is now she's there to then just make a whole bunch of daughters off of, and then that's what's used in the commercial operations. So we'll sell a breeder queen to these commercial queen producers. They'll make thousands and thousands of daughters, and then these daughter queens are then sold commercially to all the commercial beekeepers in the US. So going back to before, only 473 queen mothers making up all 1/3 managed colonies genetics, we can widen that a lot by supplying commercial queen producers with more genetics, more subspecies, etc. So over the years, WSU, the bee program here, then the Shepherd Lab, now Hopkins Lab, has made several semen collecting trips to Italy, to the Caucasus Mountains, to Kazakhstan, to Slovenia, to all kinds of different places, to the country of Georgia. And, they've been able to do really quite a lot. And in this image here, we have queen producer Jackie Park Buris, who's a very widely known queen producer in California. She's known for producing Italian queens, and her husband came with on this trip. to Italy to get more Italian genetics. So that was pretty cool. And I don't know, it's cool to see the different styles of these hives that are used there. And here's a very scary picture of their trip to Mestia in the country of Georgia. And here's what those hives look like. So really exciting stuff. I really hope to one day go on a trip like this to collect some semen. That would truly be amazing. And most recently, just in 2023, Brandon and Steve and Megan Mahoney from Queen Producer in Texas, and a group of others, and Sue, all of them together, they went to Slovenia to get Slovenian bee genetics. And they have these cool huts where the hives are kept in the wall and they pull the frames outwards towards them. It's pretty interesting. And another notable one would be in 2015, bringing in semen from Apis mellifera pulmonella, which originates in Kazakhstan. And Kazakhstan, if you didn't already know, is the area that apples are thought to have originated. So kind of the thought process behind bringing in this subspecies that's native to or endemic to that same region would be, hey, maybe if we brought this in the US, it could potentially be really efficient at pollinating apples, which would be really relevant to our Washington state agriculture. And of course, Steve named it Pulmonella after Pomona, the goddess of fruit. Yeah, and in 2023, a graduate student who was here at WSU, Adam Ware, he was able to get these genetics from the semen from Kazakhstan into some live form here. And you might be wondering, if the semen was collected in 2015, how did he do this in 2023? Well, this is how. So Brandon Hopkins, who is my advisor, when he did his PhD here in this lab, he was the one who developed the methods and protocols for cryopreserving honeybee semen. So this like really opened up the limitations, because before, you know, they would have to go make a trip to get semen every time and then immediately inseminate when they got back, because that stuff's only going to last like 2 weeks or something. But being able to cryopreserve semen means that we can store these different genetics here at WSU for, you know, theoretically hundreds of years or more, and we would always be able to revive it. And of course, cryopreserved semen is not going to have as good viability as fresh semen. Something that's interesting is bee sperm has really, really long tails. If you look at it, I should have put a picture in here of that, but compared to like mammal sperm, there's really, really long tails. And so kind of as a result, getting frozen like that, there's like more opportunity for all these tails to get like broken. But anyway, regardless of if some is unviable, we're able to preserve these genetics, and that's what's important, because we can at least get that into a live form and not have to make so many trips back and forth. So over the years, WSU has brought in genetics mainly, you know, from all these different places, but kind of the main notable ones were the Carniolands, the Caucasians, and then of course the Italian honeybees. And this was not just something that WSU was able to just do right away. This is something that actually required a lot of approval. And WSU and the USDA, as far as I know, are the only entities that are able to do this, that are like federally approved to import semen. And kind of the main reason we were able to get away with that was Steve Shepard. He was arguing, well, why can't we bring in this semen? And finally, kind of the agencies were like, you can if you do it on an island, if you're able to breed these bees first on an island where they are isolated, and we know they're not going to be spreading whatever potential disease or viruses could be transmitted through that semen to other honeybees around, and we need to make sure that they're safe first. And Steve was like, okay, well, that's not going to work. You know, we're 7 hours away from the ocean. How would we do this on an island? But we had this amazing spot out on Smoot Hill, which is in Albion, just like 20 minutes out of town here, just north of Pullman. And he was able to argue that we were an island in a sea of wheat, and that we were isolated enough in this area, isolated by agriculture of the Palouse, that we could do it. We could have semen bred into livestock and have that actively going on here and be able to observe if there were any viruses or anything before selling these to the queen producers and such. And it worked. So we don't have any bees out here right now, but we still have the space out on Smoot Hill, and it's beautiful out there. I hope we do have bees out there again one day. And now, another cool thing, well, it was a while ago now, but kind of a big deal is that WSU was able to get some of these straws of semen cryopreserved and stored in the germplasm in Fort Collins, Colorado. And this USDA facility that they have there, they store different, like, not just semen of different animals, but like different, just preservations of different genetics, different plant tissue, all kinds of different tissues and things that they store here long-term for the purpose of if there was a nuclear war and well, I don't know if that's necessarily the purpose, but that could be one idea, right? Is if something happened and everything was destroyed, how could we revive genetics of organisms we had before for livestock or for whatever purpose? Another idea that's maybe less extreme would be, what if 100 years from now, people go, oh, those Jackie Park Burris Italian queens were really good. They were top notch. They performed so good. I wonder if we could bring those back now. So anyway, there's all this genetic material being stored here, and with being able to import semen and get that approved and making these trips, WSU was able to contribute by having some of this honeybee semen stored in Fort Collins. So that's really special. So with that, I hope that this makes sense of how this is so impactful to apiculture as a whole, and how, if we were not able to do this, if we were not importing semen, we would have very, very limited genetics here in the US. And yeah, we would lose a lot of potential, that's for sure. We would be limited to those 3 subspecies we originally had, Ligusica, Carnica, and yeah, whatever the first 3 were, I forgot already. But something that a lot of people ask me when I share about this is, well, what could I do to learn instrumental insemination? And I think it's a great thing that people really want to learn it, but there are some things to consider. Mainly just that it's such a specific tool that you really, really have to have the rest of your toolkit ready to go in order to use. So like I mentioned with how intensive it is to first have drones and then have virgin queens ready to go, you need to be able to have a good population of colonies to pull all these resources from. So if anyone out there listening is like, this is really interesting to me and I really want to learn instrumental insemination, I think that's great. And what you can do to get there is first become very proficient and familiar with queen rearing. Because if you're not able to rear a whole bunch of virgins to have ready to go to inseminate, then how are you going to inseminate? And with that, kind of going along with that, is just having enough colonies that you're comfortable managing year after year and that you're able to draw resources from. Because every time we're banking drones and every time we're banking queens, we're pulling brood and bees and other resources from your general population of stock. So if you're interested in it, I would say make this your goal if you are not already able to do this. So start rearing queens and start managing a good population that you're able to pull resources from. So with that, that is my presentation. I think I ended up a little early, but I would like to close us with a quote from this book that recently came out, Honeybee Genetics and Breeding by Robert E. Page, who's one of the original guys who developed instrumental insemination methods, and one of Sue's original teachers, which is how she originally became such an expert in this field. So this is a quote from the foreword written by Sue. Honeybee breeding is a lifelong adventure and passion. As a young idealistic beekeeper venturing into developing a commercial stock, Dr. Laidlaw told me, you will never be bored by the challenge of selecting the super bee. She doesn't exist. The journey will be the reward. 4 decades later, this has proven to me to be true. So keeping that in mind, with all of our potentially have with insemination, I hope that people will continue searching for the super bee. And maybe she's not out there, but maybe she is. And hopefully we'll find her one day. um, but yeah, I am happy to take any questions anyone has. And if anyone else has any further ideas or thoughts they would like to share with me, I have my email here and feel free to reach out to me.
Thank you so much, Molly. We've been struggling getting the live going, so I'm just going to ask a handful of questions I came up with, and then hopefully we can have you interact with our Facebook group and our YouTube page to answer any of those follow-up questions that get posted over the next month or so.
Sure.
Um, can you kind of go into just the simple or the important version, if you didn't cover it already, a little bit more intentionally about why the importance to bring in new genetics and kind of what that's going to do, especially in this age of such intense pests and diseases, or what
Yeah,
we're hoping to learn from that.
totally. Yeah, well, genetics in general are just, they can be so different depending on where you're getting them from. So I think just by having wider options, casting your net wider of having more genetic diversity, you're more likely to see something that will, how do I put this? You're more likely to see different traits arise in different situations, right? So if we can have a really diverse pool of like mother queens that people are selecting stock from, and we're selecting, based on which of those mother queens are producing the most hygienic bees, we can maybe see more options for bees that would be more hardy to different pests and maybe just able to survive them better in different ways. Um, and that's not only just based off of like what genetics we're actively bringing in, that's also going to be what new genetics, the further generations of those first generations could be. So like, for example, we could bring in some new subspecies that maybe it performs against varroa the same way other subspecies do, but we could cross it with something else and we wouldn't even know what the possibilities could be. It could be something even more robust. But just having like diverse genetics in general is like a necessity of fitness. So let's say theoretically we only ever had that one subspecies brought in and we just kept it as that, we might see very, very reduced fitness in our bees by now after hundreds of years of breeding from the same original few colonies that were brought over.
On that note, with a few colonies, can you share kind of, in a continuum of 0 to 100, what the potential um, perspective of what the colonies that were migrated over, because that stopped in the early 1900s, 1920s, I believe, kind of how much extra diversity is out there that we have not even tapped into. I mean, we only brought in 3 major subspecies. What else could be out there from a genetics perspective?
Yeah, that's a great question. So let me go back to this map here because I think it kind of illustrates it pretty well. So this is showing all the subspecies of Apis mellifera that are recognized. Many of these, I had not even heard of before because we don't have them here at all. Like some of these from the southern parts of Africa. So really, like I would say the genetics we have here in the US are going to be, even with us bringing stuff over from these specific areas, like when we brought the Caucasians in, even after all that semen importation, there's still so many other different subspecies out there. And there could be even more subspecies that like aren't recognized yet. Um, but yeah, I mean, it'd be interesting if we did bring over more of these from other parts of the continents. That would be interesting.
What about, so we hear a lot about, I'm just going to throw out there for new beekeepers, there's ankle biters and Saskatraz and Russian bees, the standard 3 that we know of, Caucasian, Carniolan, Italian, very popular, at least here in the Northwest. Um, are those kind of subspecies made of these primary species that were initially brought over, or are we starting to see new subspecies from some of the work that WSU is doing?
Yeah, that's a good question. I'm not familiar with like each one of these lineages, but I know like the Caucasian bees that we have here in the US are going to be sort of a mix of the Caucasian genetics we brought over and the Caucasians that could have already been there uh, before that. Um, yeah, and I'm honestly not super sure. I think that there are other labs looking into like fully genotyping the different subspecies and like going back in like historical specimens and like comparing that to them. Um, but yeah, I don't know if I can say much more about that. It would be interesting to know more.
That's okay. I just wanted to challenge you a little bit. It looks like from the map, we can see, right, the African portion of it. Some of those may not be suited to the geographics of the United States. So
Mm.
they may or may not do very well here if we did try and bring those genetics in. Switching gears a little bit just to get your perspective. So you talked about that raising bees in that sea of wheat, and regardless of the University of Washington's primary location in Pullman is still surrounded by a sea of wheat. How is it to have your host colonies that you do a lot of research of in that sea of wheat without a lot of natural forage? Because it's still a very high desert, dry summer climate.
Totally. Yeah. So that particular area, I think, was a very specific, like, we're pretty darn isolated in wheat. So I think a lot of our colonies will have a lot of other things to feed on. So, like, for example, we do have a lot of hills with canola growing around here, and I know a lot of our honey ends up being canola honey as a result. But yeah, like in areas like this in Pullman with a lot of like cereal or like wheat different grains being grown that pollinators might not forage from, there really isn't, it is very sparse. So, like a lot of other places, we are supplementing our bees diet with sugar syrup, not throughout the whole year, but in the early parts of the year before the nectar flow really starts, and then in the end of the year when the nectar flows over. So we do supplement a lot with sugar syrup during those times. And other than that, like our bees are eating a lot of canola, and black locust is kind of our other main one, I think, around here. Um, yeah, and maybe this is something someone else has already talked about on here before, but another thing our lab is looking into is developing these patent pending products that this Belgian company is trying to get going. They're pollen patties that would theoretically completely supplement bees diets for the need of pollen. Not that we wouldn't want them foraging, but just for situations like this, where for a good part of the year, there's nothing really blooming and nothing really for them to forage on. Um, so that's kind of an interesting thing to combat that.
We think about forage and just really focus on the nectar, but your point on pollen is on par with especially raising queens and pollen and protein quality being a key. How is the lab finding queen rearing, especially after that primary pollen flow going in the later part of the year?
Yeah. Uh, pollen is like, yeah, kind of the main important thing with queen rearing and everything. Um, And honestly, yeah, I think the answer to that is just we're very limited for the time of year. So, for example, right now it's getting to be too late to be making cell builders and grafting. Um, and yeah, we only really are doing that probably between like May, June, and July usually. But that would be kind of an interesting thing to look into would be if these pollen patties were working to develop could potentially how much of an effect they would have in a queen rearing situation. That would be interesting.
That could be really important for those early season queens that sometimes don't seem to do well if they're a little too early, little wrong climate.
Yeah.
So this kind of new world carniolan that's been worked on, has anything vastly different been seen in some of the studies in the artificial insemination that's been done generation after generation. I know they're out there in the marketplace being sold by queen breeders. What is so special about the work that's been done on this queen or this line?
Well, I think a lot of care has been gone into like maintaining genetic fitness. I know because Sue is sort of the one who did all that with the New World Carniolan. And from what I understand, I don't want to speak too confidently about this in case I'm incorrect, but I believe the New World Carniolan is a mix of Old World Carniolan genetics combined with traits that have been selected from US populations that are similar to that of the Carniolan. So I guess I'm sorry, I'm kind of forgetting what the original question was, but I guess I can't answer too much about how different it would be, but I know Sue, kind of her main thing is if we are going to breed after so many generations, making sure that we're maintaining that fitness and not narrowing ourselves in too much.
One of the things that backyard queen raisers or backyard beekeepers don't seem to do a lot of is being intentional about trait selection. You see a queen cell, you split it off, right? You see a potential swarm happening, you do an emergency split. For those that are managing multiple colonies, can you go through kind of maybe what the things to look for to make a really good split to help build up someone's apiary, what they really want to do to help improve the genetics. And obviously there's some personal preference there, but just kind of some general things of potential considerations.
Yeah. So, from what I've been taught, one of the main things that comes to mind is checking for brood pattern. And specifically, I've heard Sue talk about this in the context of when we're breeding multiple generations from the same stock, like kind of just keeping an eye on the brood pattern to make sure we're not going too far in and kind of like crossing the same thing too much. Because brood pattern in general will be a great sign of like overall fitness of the queen. So we want to see like a good laying pattern with like full brood. And usually, we want that sort of layered effect where we're seeing eggs, older larva, older, and then like pupae, capped brood. And then other things, of course, to look for would be how hygienic are the bees. So I was going to say a lot of queen producers, but also people who keep bees as hobby and rear queens as a hobby will do this test with liquid nitrogen. I'm sure many out there watching this have already seen this, but they'll take like a can and squish it over the frame, a frame that has a bunch of capped brood, I should say. And then they pour liquid nitrogen into that can, and basically that freezes all the brood inside those capped cells, killing them. And then you can remove it, put it back in the colony, and over time observe how much are the nurse bees and the other worker bees going to clean out those cells and recognize that the pupae inside there are dead. So having that hygienic behavior is going to indicate a lot of fitness in terms of like being able to fend off parasites or viruses and just being able to overall like maintain health of the colony by removing debris. So I would say those are two main things would be hygienic behavior and brood pattern.
And
And of course you can score other things as well, like like quality of feed, how much a lot of times we'll be scoring things on a scale of 1 to 3 or 1 to 5, like how strong is this colony food wise? How much food do they have built up? And just something simple like that on like a simple scale.
how much honey they produced in a season compared to others. Um, Thinking of things that, some backyard beekeeper might want, how nice they are, not mean.
Yeah,
How much
temperament.
food did they consume over winter compared to their peer group in your apiary? How strong do they come out? Are they very swarmy and you have to manage them very early, or can you have a more relaxed tone and wait for their buildup when the weather is more acclimate? Back on the hygienic behavior, liquid nitrogen is not readily available by, just the can't go
Yes.
to the drugstore or the hardware store and buy it off the shelf. Is there an alternative method that someone might be able to test their bees' hygienic behavior. Maybe it's in the mite wash testing over, kind of do a bi-monthly or monthly mite wash test through the main season, or
Yeah, absolutely. The mite wash would be an excellent way to just test it. There, I imagine there probably is a way that people just in their yards do like some other way to freeze is what I'm thinking other than liquid nitrogen. But yeah, I'm not sure what that would be.
there's
But yeah, the mite test.
there is a wart remover that is liquid nitrogen. I don't know if you can get it out in the concentration you would need to get a big enough patch of brood, but so I'm not recommending that. But we might want to follow up on that one. That one could be fun.
Yeah, that would be interesting. I just thought I'd pull this up real quick. I meant to shout him out at the beginning of this presentation, but a lot of these images and just the slides in general, I tailored from Steve Shepard's slides that he gave me. But he had this slide that I thought was really interesting that I pulled up real quick. So here's what he wrote as what we would use to evaluate colonies throughout the season. So how well they're able to overwinter, and then their temperament, like you mentioned, and that would be something I know we would score on like a scale. And honey yield, same thing. And you mentioned how much honey they have at the end of the season. That would be valuable, but also in the field when we're just doing our colony checks, even not in a queen rearing situation when we're just like checking and we have like a sheet we're filling out, we will just see how's the honey yield doing right now? Like, does this feel medium, heavy, or light? And that can just be kind of a baseline thing to keep track of whenever you're doing your colony checks. And then, of course, hygienic behavior. And then, and disease resistance. So, yeah, just avoiding any signs of disease. And then spring buildup. So, how quickly they're building up that population after the overwintering time.
Awesome. That's all the questions I have off the top of my mind. Again, if you could come back and monitor those chats for at least the next month, that would be amazing. Any last things to add on your end?
I don't think so. Yeah, thanks everyone for listening. And yeah, again, I believe I'll be back here in October to talk about the research using entomopathogenic fungi to develop a biological control for varroa mites.
We look forward to it. Thanks again, Molly. Really appreciate it.
Of course. Thank you, Courtney.